Thank you very much. For our second speaker, we have Dr. Nallasivam Palanisamy, and he is the Associate Scientist Professor at the Department of Urology at the Henry Ford Health System, and also Associate Professor at the Department of Pathology at University of Michigan. And he has an excellent presentation for us today, looking at the combined immunohistochemistry and RNA in situ hybridization to elucidate tumor molecular heterogeneity in prostate cancer. Thank you very much, Dr. Nalla.
Hi, everyone. I'm Nalla Palanisamy. Thank you for joining the presentation. Today, I'll be talking to you about the application of combined immunohistochemistry and RNA in situ hybridization to understand the tumor molecular heterogeneity in prostate cancer. I don't have any disclosures to make. The outline of my talk is going to be, I will present some data showing the comparison of RNA in situ hybridization and protein expression, how it is comparable, because traditionally people use IHC, but I will show you how comparable both RNA and IHC, since we are going to do both IHC and RNA is simultaneously on the same tissue sections. Then, since this presentation is going to be mainly on prostate cancer, but it is not limited to prostate cancer, whatever the application that I'm discussing, it can be applied to other cancer type as well. So most of the example that I will be showing is from the prostate cancer. So with that, I will show you how we can actually detect these different AIDS family gene fusions in prostate cancer. Then using this combined immunohistochemistry and RNA in situ hybridization, I will show you how we can actually identify distinct molecular subtypes of prostate cancer, because prostate is a multifocal and highly heterogeneous tumor. So using this approach, we were able to identify distinct molecular subtypes, which I will show you a few examples. Then I will show you additional examples how we can do this assay both on a different type of tissues like tissue microarrays or radical prostatectomy tissues or the whole mount tissues like that, to study the in situ evaluation of tumor heterogeneity in prostate cancer. So I was one of the early adapters of this technology, because when ACD Bio introduced the RNA scope method, I think early in 2011 or 12, I picked up this and I was able to adapt this technology in our research. So based on my experience, I would like to share with you some useful tips. I think this will be useful for anyone who want to set up this technology in your laboratory. So basically, if you are doing the RNA in situ hybridization, I would recommend use freshly cut sections of FFPE tissues. Even if the sections are cut, make sure they are stored at -20 or -80 degrees. But I would prefer make sure the blocks are saved at cold temperatures. Then whenever you need the slide, just cut fresh so that the RNA quality will not be compromised. And so any slides that is older than 3 months, it's not recommended. Still, it may work, but the intensity of the staining, it may not truly reflect the extent of the expression level of the target that you are interested in. Then another thing is important is always test your tissue using some positive control probe. Make sure the RNA quality is intact. So there are good number of positive control probes are available. So now the technology has evolved. So now you can do both the method by both manual and automated methods. The RNA and IHC both can be combined by using either Ventana or DARCO system, I believe. I have experience in using the Ventana system. You can do both automated and manual methods. And if you are doing multiplex assays, make sure that first test the individual probes, make sure they are working first, then before engaging in the dual IHC or the dual RNA approach. And the other important point that I would like to make is always perform RNA-ish first, followed by IHC, not the other way around. And if you do the RNAs first, there is no additional kind of antigen retrieval step for IHC. So whatever you do for RNA-ish will be sufficient for IHC as well. And there is a new approach. You can do both the DNA fish and RNA-ish on the same slides. I may not have time to show examples on that, but this is a new kind of approach that we have developed in our lab. So if you're interested in looking for any, say, for example, particular gene that is amplified at the DNA level, if you want to verify whether the amplified gene is expressed at the RNA level, you can combine both the DNA fish and RNA-ish. That can be done. And another important point is that you need to have some prior knowledge about the localization of the target markers, whether it is I think particularly for IHC, whether it's a membrane or nuclear or cytoplasmic. So if you're doing like combined IHC, make sure that these markers are not expressed at the same localization. So it may be difficult to interpret the results. I think these are some of the important tips. I thought it will be useful to the audience if you are setting up this technique for the first time in your lab. And additional technical help, you may get it from the ACD Bio technical team. So first, I will show you quickly how we can detect this different type of EDS gene fusions in prostate cancer. As you know, the TMPRSS2, ERG, ETB1, ETB4, and ETB5, these are the well-known chromosome translocations in prostate cancer, and these genes express only after the chromosome translocation. Otherwise, there won't be any expression in the benign or prostate tissue without translocation. So I will show you how we can detect in a multiplex method. You can detect the EDS gene fusion. So here there are examples here. These are the ERG, SPINK1, P10, ETB145. These are the well-known prostate cancer biomarkers that can be that we have used in our research to evaluate at the in situ level. So here there are some non-coding RNAs also. Recently it was described like PCAT1, SLAP, KLKP1. So when you have these many markers, sometimes you may encounter with a limited amount of tissue, so you may not be able to perform all these markers if you do it individually. So in order to screen the incidence of these markers in a given tissue, we developed this the dual IHC and the dual RNA-ish approach. Like for ERG and P10 or the ERG SPINK1, we have a good antibody, so that can be done with the IHC. ETB1, 4, and 5, there are good antibodies in the market, but none of them are specific for cancer. It may work on the Western, but they are not good for tissue. So that's why we decided to use RNA-ish. Then the other markers, PCA3, PCAT, SLAP, KLK, these are all the non-coding RNA targets that have an important role in prostate cancer. So we can do RNA also. So, and finally, I will show you how we can do both ERG, SPINK1, ETB1, and ETB4, all 4 markers on the same slide in a sequential hybridization of both RNA-ish and IHC. Okay, first I will show you quickly how RNA-ish is comparable to IHC. Okay, so here is first we did the control probe. We can use the Pol R2A and just make sure the RNA quality is intact. So here is an example that I want to show you. So there is a tissue that's positive for the ERG protein expression, which was confirmed by IHC. Then we took a consecutive slide. Then we performed RNA-ish on the slide. As you can see, the RNA expression is only in the cancer, not in the stroma or in the benign tissue. So this clearly shows. Another important thing is you can do the RNA-ish under bright field. Although you can do it under fluorescence, but you can. I think most of the applications you can do it under bright field, so that will preserve the tissue morphology and everything. So you can read the RNA-ish data like IHG. Okay, so I think by doing this, we are very much convinced that RNAs can be extensively used. So we published on this work to come up with a different scoring criteria depending on the intensity of the signal, because each RNA transcript is going to appear as a distinct punctate dots. So based on that, the intensity, you can classify them into different categories. So now I will show you how we can detect the H gene fusion prostate cancer. So here is the list of all the gene fusions that are originally discovered in prostate cancer that identifies about 50 to 60% of the prostate cancer. Because none of these gene fusions are associated with any particular Gleason grade, so therefore the Gleason just identifying different Gleason grades may not enough to tell you what's going on. So therefore, I think the molecular screening is a new approach that is being used. So it is not only just identifying gene fusions. There are actually some preclinical studies shows the therapeutic potential of these gene fusions, which are listed in the right column. There are different drugs that are being developed, although they are not readily available for prime-time clinical use, but that can be used. Eventually, when these drugs are available, then it becomes essential that you need to screen this tissue for the presence or absence of the gene fusions. So here I will show you these are the different probes that are originally designed by the ACD Bio team, because they have the proprietary algorithm that they will design the probes. Once you give the sequence of the gene of interest, then here we initially tested the sensitivity and the specificity of this probe. Here we used the ETV1 and ETV4 and ETV5 probes because these are all H family genes and they are closely related. There is a sequence similarity on this family of genes because there are 27 H family genes. So we want to make sure that these probes are specific for each gene. So we were able to show that the probes are positive only with the right samples. Like ETV1 probe is positive only in the ETV1 sample, but not in ETV4 and ETV5 positive samples. Like that, in the bottom I show the ETV4 positive is strongly positive in the ETV4 positive samples, but not in ETV1 and ETV5. So we were very much convinced that the quality of the probes and the performance of the assay. So we decided to do a systematic screening of the prostate cancer. So this is the kind of logical diagram that shows how we can systematically screen the prostate cancer biomarkers. Because if you do it individually, like one slide for ERG, other for SPINK1, ETV1, ETV5, it will be time consuming and you may not have enough slides to do it. So I think based on this, we developed this combined approach. In this slide, I'm showing you good examples of the ERG, SPINK, and ETV1 and ETV4 positive cases on the needle biopsy. The ERG and SPINK1 is done by IHC, ETV1 and ETV4 by RNA-ish. So you can appreciate the specificity and the sensitivity of the signal. There's no non-specific signals in the stroma or the benign tissues. So this is another example to show even a tiny tumor on the needle biopsy that clearly picks up a small tumor foci that is positive for ETV1. So you can use the needle biopsy, or you can do it on the the tissue microarray as well. So in such a large TMA, we were able to identify only these 3 positive cases for ETV1. So you don't get any non-specific or background signals in any of the negative cases. That shows the sensitivity and specificity of this assay. And also we validated on a human xenograft tissue that is positive for ETV1. As you can see, these probes are actually hybridizing only to the human tissue, but not in the mouse tissue. That shows the specificity of this probe. So we actually published this paper. Anyone who needs more information, please go through this paper. You'll learn more about the application of these probes in your research. The next, I will show you how we identify new molecular subsets of prostate cancer using the combined IHC RNA-ish. So here I will show you an example. This is a radical prostatectomy tissues, the H&E tissue. And this is, we know that this case was positive for ETV1 by doing FISH analysis, because at that time, RNA scope was not available, so we have to do either PCR or FISH. So we know that this case was positive for ETV1 rearrangement by FISH. So we did the RNA in situ hybridization using the ETV1 probe, where we found that majority of the tumors are positive with the ETV1 signal, but there are small areas, and on the circled areas, they are negative for ETV1, but still they are cancer, okay? So out of curiosity, what I did, we did another probe like the ERG, immunohistochemistry on a consecutive slide. And interestingly, what we found was the area that is negative for ETV1 is also positive for ERG. So this clearly shows the intra-tumor heterogeneity, so the because morphologically they all look the same, but at the molecular level, they are clearly distinct. So just by doing ETV1, you may call it as just ETV1 positive case, but we clearly show that there is a ERG small tumor that carries ERG as well, and we confirmed both ETV1 and ERG by doing FISH using the specific probe for ERG and ETV1 and confirmed the rearrangement. Okay, so this is the first evidence that we started, because until this work, people are thinking that each tumor contains only one specific gene rearrangement, but this is the first evidence we show that there are more than one gene fusion can be present within a single tumor. So this is another example where you can show two adjacent foci, like one is positive for ERG, the other one is positive for SPINK1. In this one, so this high-grade pin strongly positive for ERG, and a foci right next to that is positive for SPINK1. So these are all mutually exclusive. There is no overlap within the same tissue. So this clearly shows that the origin of different driver mutations in the different tumor foci. Okay, so this is the first evidence that we clearly show that the prostate cancer is highly heterogeneous and more than one molecular marker can be present. So here is another example. The ETV1 is a large majority of the tumor is positive for ETV1. And at the bottom, there is a small foci that is positive for ERG, and still there is a small area that is still negative for both ERG and SPINK1. So there is a third tumor. We don't know what is the driver mutation going on. So this clearly tells the enormous complexity or the genetic heterogeneity of the prostate cancer. And we are right now we are working on trying to understand what is the influence of this type of tumor heterogeneity in the overall clinical progression of the disease, whether it makes the tumor more aggressive or indolent, or how aggressive these cancers are in establishing metastasis and other one. So now I will show you quickly the application of combined IHC and RNA-ish to unravel the tumor heterogeneity here. So as I showed you before, you can do the RNA-ish and IHC both on the needle biopsy, radical prostatectomy tissue, and the tissue microarray. What we found was it is not always the so-called the dominant nodule in the prostate cancer, because it was the general belief that the dominant nodule is the one driving the biology of the disease. But recent work that clearly showed that at least 15 to 20% of the cases, even the secondary foci is also involved in establishing the early or lymph node metastasis. So in that situations, although about more than 80 to 90% of the prostate cancer are highly heterogeneous with both the dominant foci and the secondary foci. So it is important to evaluate all the tumor foci, then get a combined kind of score, what is going on in terms of actually driving the disease. So keeping that in mind, what we found was instead of just selecting only the dominant nodule, we don't want to kind of bias with any particular small tumor. So we decided to do the whole mount tissue without actually taking any particular tumor. We wanted to evaluate what is going on in the entire prostate, whether if you have a multifocal disease, whether all the tumors are carrying the same mutation or it is going to be different, okay? So this is what we are talking about. So the tumor heterogeneity, because the multiple tumors are occurring or developing at different time point, and they are at the different stages. Some are high-grade pin or adenoma, so like Gleason 6 or 7, all those different type of tumors are there. So our goal is to understand what is the genetic relationship between these tumors. So for that, there are some preliminary work that people have done by doing the sequencing analysis and other things. So when you do the sequencing, you are just crunching the whole tissue. It is like trying to identify what is a fruit in a smoothie, right? It is very difficult to pinpoint what exactly is happening in the tumor. So our goal is to preserve the tissue morphology and to do the heterogeneity analysis. So in that, so initially we did a work on biopsy tissues where we selected only the biopsy cores that contain the discontinuous foci, meaning that when you have a long core on both ends there are two tumors, and we did the both ERK and SPINK1 on that, and we were able to show that both tumors are either different. One is ERK, another is SPINK1, or it is one is positive for ERK, another is negative, like that. We were able to show like about 30% of the tumor had like at least one SPINK1 positive and ERK SPINK1. So we can demonstrate this heterogeneity in the needle biopsy itself. So once we know that this heterogeneity has some influence in the driving the biology of the disease, so it is important to screen these tissues for these markers. So here again, this is the enlarged image of a biopsy where you have two tumors in a biopsy core. Like this tumor is positive for ERG and the other is positive for ETV1, like that. So you can demonstrate this heterogeneity on needle biopsy. But what we did was, so since this approach is very powerful, what we did, instead of doing only dual IHC or the dual RNA-ish, we kind of perfected this approach for the simultaneous evaluation of at least 4 markers in a single hybridization. So we can do the ERG and the SPINK1 by IHC and ETB1, ETB4 RNA-ish. So here I'm showing an example. This is a tissue microarray where we have all 4 positive samples. Like you can identify both like ERG positive, SPINK positive, ETB1 and ETB4 positive. So the one important point that I would like to make here is in prostate cancer, these markers have a mutually exclusive pattern. So if a tumor is positive for one marker, you don't find the second marker in the same tissue. So this may not be the case in other cases or another marker that you are working on, but this is very clear in our approach that we can see the distinct mutually exclusive expression pattern of all these 4 markers on the same slide. So instead of using 4 separate slides, you can actually demonstrate the positivity in a single hybridization by combining IHC and RNA-ish. So this is another large kind of tissue microarray where we did both dual IHC and RNA-ish. There are many cases positive, which I clearly show here. This is the ERK positive, SPINK positive 1, ETB1, and ETB4. So very clear, very distinct kind of staining. So here you may see some green staining. So that is because the ERG is not only hybridizing to the prostate tumor, but also it stains some endothelial cells. So we just use it as an internal control for IHC. So that's why even in RNA-ish, you will see this ERG in the endothelial cells. Otherwise, it is not anything very significant. So this is very clearly to show that how specific and how sensitive this approach is you can do. And when you are in the tumor that are negative, you don't see any staining at all. There is no background low level. So you're not looking for differential staining. Here, mostly for the prostate marker, they are yes or no, whether it's there or not there, like that. You don't see any cross hybridization. So then, like I told you before, since we demonstrated the expression of these markers, the needle biopsy, so we wanted to study the tumor as a whole. So we took the whole mount prostate tissue samples. So here we are not selecting any particular tumor here. The entire tissue is mounted on a 3x2 slide. So we can perform both IHC and RNAs on the same slide. So here, I'm showing you here very clearly. So here at the bottom, as you can see, the tumors look all the same, like a Gleason 6 pattern at the molecular level, the one section of the on the right side, you will see they're all positive for ETV1, and on the left side, they're positive for ERG1. So this is more like a collision of two tumors. They are sitting right next to each other. But under H&E, they all look like one tumor like that. So I think these are all the very important information that you can gather by doing this combined IHC and RNA-ish. And here is another example where we show that there are 5 tumors in this case, and we demonstrated both ERG, SPINK, and ETV1 in 3 different foci in the same patient, and still there are 2 tumor foci. We don't know what exactly is going on. And importantly, the so-called the dominant foci is negative for all these 3 markers. So all the marker positive tumors are in the secondary foci. Okay, so that's why just analyzing only the dominant foci that may make that you are conveniently ignoring some valuable information that might help the patients like that. So this is the advantage of both doing the combined IHC and RNA-ish. You can identify distinct molecular subsets of prostate cancer. So this is important because although the ERG and ETB1 are belong to the same kind of H family genes, they behave entirely differently under the molecular mechanism, because ERG negatively regulates the AR signal program, whereas ETB1 it cooperates with the AR signaling like that. So as I showed you before, there are some small molecule inhibitors being developed for the ETB1. So when you have this type of behavior of the different gene fusions having different influence on the tumor progression, so it is important to have some drugs that is specific for each gene fusions. So those are all some of the preclinical work is going on. I think by the time when these markers, when these drugs are available for clinical use, I think screening of these tumors with these markers will become very invaluable. Okay. So it is not only, like I showed you, is not only for prostate cancer. We have done some work on some of the sarcomas, and also we can detect some virus as well by using this RNA in situ hybridization. So anyone is interested, please go through this paper. So that's that clearly shows the wide application of this technology. So in summary, I would like to conclude showing, I think I convincingly showed you how powerful this RNA-ish approach combining with IHC, right? So this is a novel approach for detecting H gene fusion in prostate cancer. But like I said before, any marker that you're interested, particularly if you have a gene for which if you don't have a good antibody, I think RNA-ish is the best approach to use. So by doing the dual IHC and RNA-ish, we were able to identify new subset of prostate cancer. And also we showed the mutually exclusive expression pattern of these different prostate cancer gene fusions by using the whole mount tissue. And I think the distinct, like I mentioned, the distinct biology of the S gene associated prostate cancer suggests that each disease class may require new therapies directed to underlying programs controlled by the S genes. So I think the combined IHC and RNA-ish is a novel approach to screen molecular operation prostate cancer. So importantly, you can do all these at the bright field, So like I said, although I didn't have time to show some examples, so combined DNA-fish and RNA-ish can be used if you're interested in studying DNA amplifications and the associated gene expression. You can combine both DNA-fish and RNA-ish. Okay, so I think with that, I will conclude my presentations. I hope I have showed you enough data to adapt this technology. If anyone is interested, please feel free to contact me or the technical team of the SED Bio. We'll be happy to help you. Thank you very much.
Thank you very much, Dr. Palanisamy. That was an excellent presentation. Really appreciate you taking the time to go through in so much detail. That was really wonderful. Here's an opportunity, if you have not yet asked any questions, if you have any questions for Dr. Nalla, please do pop them into the chat. In the meantime, I do have a couple of questions lined up for you that we can go ahead with. Dr. Nalla, what are the advantages of using RNA in situ hybridization over IHC?
Yes, as I mentioned in my presentation, RNA is not a technique to replace IHC. RNA is a unique and technology that can be used for the genes of interest for which you don't have a good antibody. So even though there are some antibodies available for some of the genes, like different vectors have different antibodies and they may have a different kind of sensitivity and specificity, and they may have some cross reactivity and all those issues. In those situations, RNA is the best method to overcome all those issues, because RNA is a sequence-based hybridization. So you will not get any non-specific or off-target signals. The signals will be very specific, and also the way the RNA works is actually, it will hybridize only to the gene of interest, and you do not see any off-target or non-specific signals. So it is for that reason, for the genes for which even if the antibody is available, if the different antibody performance are quite variable, you can actually cross-validate your IHC data using RNase. That is one application. And for genes for which if you don't have any antibody, and if you are interested in studying the expression of this gene at the tissue level, RNA is the method of choice for the protein coding genes as well. And like I said, now the non-coding RNAs and the pseudogenes, these are the new molecular markers that are evolving, and their role in cancer is well established. So in order to study the tissue level expression of these non-coding RNA molecules, RNA is the only option right now for the in situ detection, although you can do PCR, next-gen sequencing, and all those things. Because as I showed you, because of this enormous tissue level inter and intra-tumor heterogeneity, if you want to see where these molecules are expressed within a tumor, because you are not expecting them to express in all the tissues, if you want to see the tissue specific, or then you want to combine with the morphological features, I think RNA is the best method. The other main advantage is, you can do RNA under the bright field method. Okay, so that is the beauty of it. Like IHC, you can visualize the results under bright field. Of course, there is an option to do fluorescence as well, but I think most of the time, I think bright field is the best approach, because you are preserving the tissue morphology. You can have the counter stain like a H&E, so you can relate of your expression results with the tissue morphology, what type of tissue morphology that you're seeing. So I think these are some of the advantages of using RNA over IHC. So each one has its unique advantages. I'm not saying that RNA is replacing IHC. You can still have IHC, where as long as you have a fully validated and a highly specific antibody, that can be still used. Then in place, if you want to study additional markers, you can put RNAs for that. And in that reason, I think both techniques are complementary, but RNA is really kind of sequence specific, and I think the sensitivity and specificity are much higher. For that reason, I recommend using RNAs if you have issues with any of the antibody that is available for any of the known genes. Yeah, thank you for the question.
Great, thank you very much. That was really good. Okay, I do have another one here. Can RNA-ish be performed along with DNA FISH?
Yes, like I showed you in the last slide, I think that is the other important development in this field, because FISH, as you know, that is a quite popular method, and the people are using it for detecting DNA amplifications, deletions, translocations, and small inserts and deletions and all those things. So when you do just the DNA, you will get only limited information. Suppose if you are dealing with an amplification of a particular gene, although it doesn't necessarily mean that when a DNA level copy number is increased, it doesn't necessarily mean that the gene within that amplicon is also highly expressed at the RNA level. So in order to answer that question, if you have a small focal amplicon where there are 4 or 5 genes within the amplicon, for example, so if you want to know what are the kind of, what is the expression pattern of the genes involved within the amplicon. So in that situations, you'll be able to have a multicolor DNA fish. So like, the DNA fish can be done with the 3 or 4 colors like that. You can do multicolor DNA fish. Then on the same slide, you can do the RNA-ish, because RNA signal is going to be look entirely different like the DNA fish, and you are capturing in different layers. So you'll be able to simultaneously visualize both the DNA copy number and the RNA expression. So that is the power of this. So I think that is the new development of this. Originally, RNA was developed as such, but there are new development, associated development that is coming up with that. So yes, to answer the questions, we can do both DNA fish and RNA-ish. Thank you.
Great, thank you very much. And yes, we do have a number of new items in the pipeline, so do watch this space for more information on that. Thank you very much, Dr. Nalla. That was an excellent overview of using ISH and IHC together, combined. I really appreciate you taking the time to speak today. Thank you.
Thank you very much for the opportunity. Thank you all. Please email me if you have any questions. I'll be happy to answer, or I'll be happy to help you if you are setting up this RNase procedure in your lab. Either you can contact me or the technical support from the ACD Bio company. Thank you.
Thank you. That's wonderful. And if we weren't able to get to your questions offline, please do continue to pop them in the chat, and we'll be reaching out to Dr. Nalla independently of today's session, and we'll make sure that we connect you and you get the answers that you need. Thank you very much.